B, this is one of those weird, okay, you have to assume since he's marrying into this family, and to get anywhere else with the problem, we assume that he is not a carrier. Okay. So with B, we assume homozygous dominant, not a carrier. And so still keeping on generation number 1, and for 2C, individual to be homozygous recessive, what does that tell you about her parents? Yeah, they have to be carriers. They're not showing the disease, so you automatically know they're not homozygous recessive, but they produced a child that has the disease, so they obviously carry that disease allele, but since they're not actually affected and showing in the phenotype that we're looking for, we know that they have a dominant allele as well. Okay, so these are some known pieces of information that you have. Okay, what about the children from 1A? Oops, I didn't change that one. That makes it confusing. Sorry, guys. So we'll make them be all the way across A, B, C, D. Right, kids from 1, C, and D would be heterozygous. Okay, that part's apparent. So these are known pieces of information. I told you to assume anyone marrying in from the outside is homozygous dominant in this case, not a carrier. So we can assign genotypes to those individuals marrying in from the outside. Okay, now we kind of got down to generation 2, individual B. What do we know about her?
She's too. But she's not.
Too
Inside joke.
She's
obviously-
cheating.
I did it. Oh yeah, well she's, she's around town. So okay, so excluding any moral decisions that we're having right now, what is her phenotype, genotype? Geneticists don't care, we're all like, okay, but you go to your psychologist and psychiatrist about that.
She's recessive.
So she obviously does not have the disease, so she must have one big D allele. I can't believe I didn't pick up on that the first time. I'm such a biologist. I'm like, yeah, whatever. She's going to make this one. Okay, but do we know her second allele?
No.
No, because she could be homozygous dominant or she could be a heterozygous, right? So that's an unknown, so you can put a little dash there. And that's true for everyone else on the board right now, right? Is we know...
No, kids over here are going to be.
Yeah, so the kids over here on 3E through G, which is at the point, yeah, you kind of work your way through it. You know that everybody, and so a good practice is for all, especially when you encounter an autosomal or sex-linked recessive, if it's any sex-linked inheritance pattern, I would put X's and Y's for everybody. That would be the first thing I would do, so I make sure I keep my alleles straight. Then I would assign any of the genotypes. So everybody else that's normal, since it's a recessive inheritance pattern, you can give them all that dominant allele. Okay. Hopefully I get some really juicy background stories on the exam. You guys can make up an entire storyline for why B and D are having a child. They are. Okay. So how do we start? How are we going to solve? If we're talking about an illicit, I guess, mating here between these two individuals, right? Okay, so we know the probability of 2D. We know exactly what their genotype is, right? But we don't know the genotype for 2B. So we have to start by solving what is the probability that 2B is what? A carrier or homozygous dominant? A carrier, right? Because we don't really care. If she's homozygous dominant, then it doesn't really, she would never be able to give an allele that would produce a recessive inheritance pattern, an autosomal recessive child that has the disease, okay? So if she's homozygous dominant, it's a moot point. Now, none of this matters. It isn't worth solving further. So always you're solving in terms of a recessive trait pattern. You're going to be solving whether they're a carrier or not. what's that probability? So we want to know the probability that she received this little d allele, the recessive allele, from either one of her parents. So that's the first probability that you need to solve. Okay. And you can go back to the first generation, so you have to go back up a generation. When you do these pedigree problems, you kind of move between generations to solve this. Yeah. So you're looking at, to get this piece of information, which just unfolded in 1/2, how did I magically get this number? So generation 1, individuals A and B are both heterozygous. So you can come back, either you see this directly in your head, you don't have to waste time doing a Punnett square, or you come back to your muscle memory and run the Punnett square. But you're not, at this point, you're not solving the probability that, yes, we'll get to that. Okay, I'll just answer your question as yes. Yes, there is 0 chance. But I want you guys to think about the possible alleles that can be given. Yeah, so what would you change about your answer? So we originally just thought 1/2, right? Because here, if this is individual, what we're doing right now, if you're lost at all, this is 1A mated to 1B, and there are possible gametes that they produce through meiosis, right? So what are the possible alleles that they send away during meiotic divisions, and what could they possibly give to their daughter here, 2B? Okay. And we want to know the probability that she's a heterozygote. Okay, so 1/2 of their gametes will have this, be heterozygote carriers. The problem is with running these kind of these sorts of pedigree questions is you do need to take into account any information that you know at all about the person. So 2B, we know for a fact that we know one of her alleles. So in essence, we're only solving for this second allele. We know that one of them is big D. So we know that she could be a heterozygote carrier. She could also be big D, but can she be homozygote recessive? No, right? Because otherwise we would see like a little filled-in circle. So ask yourself that question as you run through, the reason I'm drawing Punnett squares, we're going slow, so you see this process that you have to eliminate any possibilities that there's no likelihood whatsoever. If there's no likelihood, because of given information that you know that she has the disease in an autosomal recessive case, we eliminate that from the probability. So we would have to change our answer up here to now it's 2 out of 3 total possibilities that she's a heterozygote. Okay. The second sort of set of probabilities that you need to calculate. So given this, we know, where are we getting to? We know 2D for a fact. We know exactly what his genotype is. He is a carrier. So your next step, you don't have to calculate any probabilities for him because it's known. But the next step for this part 1 is to determine, okay, now what's the probability if, given that she is a heterozygote and she produces a child with this individual that we know their genotype, so now you need to solve probability of a child. This child that we're talking about, this potential one, will be what genotype? Yeah, homozygote recessive. So we want to know the probability the child is homozygote recessive. And this is between a mating of 2 heterozygotes, 2 carriers, right? So now this is like the same Punnett square that you have going on over here. Right? Only now you're making this assumption that we're pretending like this is 2B, like she does indeed have that disease allele. We know for a fact this is 2B. So what's the probability if they were both heterozygote that they have a diseased child? Yeah, 1/4. So this guy here. So So this is how I approach these problems. You may find a different way, and that's great. And you can come to office hours, and we can walk through. I have other possibilities of ways to think about these problems. But what I do is I walk through each piece of information that I need to answer this question, and then I gather up all of those ratios that we solved for. Okay? So on my page, as I make all these probabilities out, I would collect up all those fractions that we've calculated, and then now what do we do with them? Why do you multiply? Why are we using product rule? Okay, so that's the most simplistic. You're not saying either or, you're saying and. You want all of these, you want 2B to receive the little disease allele from either mom or dad, doesn't matter, and you want her to run over and have her illicit affair with 2D, and you want them both to give that disease allele here to the child. So you want these events all to happen together, so you would use a product rule here.
Mm-hmm.
So that's your final answer to this question would be multiplying 2/3 times 1/4. So 2 to 12 is 26. Yeah.
So if it said, what is the chance one of my children will have a disease if you use binomial expansion?
No, so thank you for asking that question. So there was one additional problem that I didn't get a chance to pull from your homework. I pulled a couple of the other ones that had the binomial because we haven't covered that, and for this course we're not going to use binomial. It's an extension to this basic level of genetics. Yeah, but thank you for bringing that. So if you find yourself needing a binomial when you're doing the homework, you're feel free to not do that problem for one thing, and I'll search around and find that problem again. I think Melanie brought it up to me too. So you don't need to do binomial expansion if you see that in the textbook. Okay, we're not covering that. Okay, any questions about how we solved this part? Possibly. Yeah.
So, maybe explain to me the 2/3, because
Okay.
what I was originally thinking is like she has one D allele, right? And so then from the other parent, it's a 50-50 shot, which what her second allele is going to be.
Right. Okay, so but for each, so what you're saying is we know she has one uppercase D allele, and the second allele is going to be a 50-50 probability from each parent, right? But the problem is you don't know which allele came from which parent, and so statistically you have to consider both parents in the equation. You can't just like rule one out, even though the way you just worded that, it sounds correct, and the logic follows. But because we don't know which allele came from which parent, you have to take both parents into consideration, and then that brings you to all the possible gametes that they can give. Yeah. I don't know if that helped.
I believe.
Okay. Any other? Yeah.
So are we not using the assumption that she's big D, just because that wouldn't have yielded a child with the disease? Or?
Right.
Yeah.
This option right here.
Yeah.
Yeah, it's similar but different than how we ruled out that she doesn't have the disease, so we don't even consider that in our denominator, right? But if she's big D, could she ever produce a child that has the disease, not in an autosomal recessive case? Yeah. So we discount that option because then your answer would be 0. Yeah. Okay, let's try another one, which is actually straightforward. So what if we had I think you guys thought the previous one was funny. I'm sure you'll like this one even better. Okay, so 2D and 2E. What if they have a child? They just, no, I shouldn't say that. That's gonna name a part of the United States. Anyways, so 2D and 2E have a child. What's the probability the child will have? And I chose this one because I want to illustrate a point here. What do we know about 2D and 2E? They're both carriers, right? So the reason I'm including this one, other than because easy, I guess, they're not both carriers.
Yeah, they are.
I did, but I jumped ahead of myself. I'm sorry. I have additional questions I want to give you guys about these two individuals, but first, let's not make it quite an inbred. And do 2C and 2D. We are going to come back to the other one. Okay. So we'll keep it outside the family still. Okay. But now we have an individual with a known genotype who has the disease, mating to, I don't know, these sisters have the same friends. So mating to an individual with, again, a known genotype, they're heterozygous. So, if this was a mating between them, what's our probability that they could potentially have a child with the disease? 1/2, right? So do you need, so the reason I'm including this kind of somewhat easy problem after we worked through more calculations earlier, do you need to do any additional probability calculating for part 2? No, right? So I just wanted to include that so that you guys, it's easy to get sort of tripped up. And that's why how I would approach these problems is assign your genotypes first, so you keep all your alleles separate and organized next to the individual that they belong to. So when you reference back to the question, you can not put additional work on yourself, not start calculating in for probabilities that you don't need to. Okay? So you just automatically, with these two individuals, we know their genotypes based on. Whereas if you hadn't walked through at the beginning and assigned those genotypes, you might be giving yourself additional work by trying to solve things. Okay. Okay, let's go back to the more interesting ones. And actually, I think I will still keep it not totally brother-sister.
She did.
It's all about morning.
So part 3, let's actually go with,
Esa gente loca que
let's go with this guy.
es que Laura, you know, it's Laura.
So let's go with 3D. I think I forgot that 2D and 2E were brother sister. Okay, so more likely possibility is that 3D and 3E get married. Now, what's the probability that they have a child with these? Okay, so for both of these individuals, for 3D and 3E, we only know one of their alleles, right? So we have to walk through the probabilities again. So we have to calculate the probability, eventually you're going to be calculating the probability that 3D is a heterozygote for them to have a child with the disease, and you're going to have to calculate the probability that 3E is also a heterozygote and carrying that disease allele. And then we also have to calculate the probability that they have a child with the disease.
So would these be 2/3 again?
No.
No.
And we'll see why here in a second. Yeah, so let's just start with probability that 3D is a heterozygote, is a carrier, okay? So we look back at 3D's parents, okay? And since 2A is marrying in from the outside, we know for a fact that they're big D, right? But 2B, we don't know, right? All we know is that they have one big D, and we have that, what was our probability that they were a heterozygote? 2/3, right? We calculated earlier that this is a 2/3 chance that they are carrying that disease allele. Now, for us, again, for this assumption to keep going, though, which is gets back to Amanda's question, for us to even answer other than giving the question a 0, there's no probability, both of these parents have to carry that recessive allele, right? So we have to make this assumption that, yes, that 2/3 probability happened. We'll take that into account, so we write down 2/3, but we're going to assume that 2B is indeed a heterozygote. So you throw in that little d that's a 2/3 chance here, so that you can calculate a probability of them then passing, put a cap under it, then to calculate the probability that they pass that disease allele onto their son, 3D. So you fill in your Punnett square, and you determine that 1/2 of the time, if 2B is a carrier, 1/2 of the time, given a mating to 2A, you would have a heterozygote child. So right now, and again, you could solve all these other probabilities of 3E. We could move on. We could do the child. You can do all this and just stack up, like I did on the previous problem, all your fractions and put them all together at the end. Or you can clump them for each of these probabilities you write up. So there's different ways to think about this. So we could now write up what the final probability of 3D being a heterozygote is, right? So we would just multiply our 2/3 by 1/2. Or you can collect these all at the end. So that part's kind of, it depends on how you think about these problems. So I'm going to move on for a probability of 3E being a heterozygote. So what do we know about 3E? Has one big D. What do we know about their parents? Yeah, one's homozygote dominant and one is heterozygote. And we actually know these two genotypes for a fact, right? So we don't have to do any calculating up here the way we just did for 2B. Okay. So 2E and 2F. Whenever their gametes come together, okay, it's a heterozygote times a homozygote dominant. So you have half of the time, right, that they will produce children that are carriers. And we don't have to add any, for this particular calculation for the probability that 3E is a carrier, is a heterozygote, we only have one fraction up here on the board because we didn't have to calculate anything about their parents. That piece was known information. What's our last probability that we have to calculate? So the actual child, right? We have to determine whether this what's the probability that the child itself, out of all the, so the probability that the child actually has the disease, because 3D and 3E could certainly get married, have lots of normal children, right? Because neither one of them shows the disease. That means they're not homozygote recessive. They must carry one of those dominant alleles. So they can have normal children. So you have to calculate the probability then that they actually have a child who does have the disease. Okay? And again, you're using your two heterozygote cross and coming up with, what's our fraction? Again, a fourth, because this is autosomal recessive. So this is what I mean by, to answer this kind of problem, however you, want to break it down, you can do that, or you can kind of stack them all up. So we have 2/3, 1/2, another 1/2, and so we have 248, right? 1/24. Maybe, if I do my math right. Okay. So, this is a really slim chance, right, that 3D and 3E could have a child with the disease. And it should make sense to you, because the more times you say, I'm asking for this probability to happen, and this one, and this one, and this one, we're getting a smaller and smaller number, smaller and smaller percent chance. What if I give you a new piece of information? What if I told you 3D and 3E, maybe we track this family, and a few years down the road, they have a normal child. So maybe this is part of this long, extensive problem here. What if we told you that they have a normal child? Now what's the probability that their second child will have the disease?
1.4.
Yeah. So why? So someone else. So the answer from this side of the room is 1/24. So someone else explain why it's still the same answer. Why did having a normal child give them no new information? Yeah. Jason. It's an independent event. And what does it tell you about the genotype of the parents? Any new information? No, because we already know that they have one big D allele here, right? Because they're phenotypically not showing it in an autosomal recessive disease. So them having a normal child still doesn't tell us whether one is homozygote dominant, the other one's heterozygote, or whether they're both heterozygote. So the same probabilities have to be calculated, okay? So this answer would still be a 1/24. What if they had a second child and the child has the disease? Now what do you know? So they're both carriers, right? Okay, for them to have a child with a disease, or you know, if that information, part 4 said they had a child with the disease, that would then give you a new piece of information about the parents' genotypes. That would tell you they're both carriers or both heterozygote. Now the probability of them having a second child, or I mean a third, I guess in this case, a third child with the disease, so for your notes. So what if they had second child with the disease? What's the probability of the third having the disease? What would your answer be? Yeah, well, now it's just a quarter, because now you know exactly what their genotype is. For them to have any child with a disease, they have to both be carrying that recessive little d allele. And that's how a genetic counselor would address this. And you can see how that's obviously on the human, ethical, emotional side, that's pretty difficult news to bear to someone to say, originally you were telling them a 1/24th chance, and then they have a child with the disease, and now it's a quarter of the time you're always gonna produce a child with the disease. Yeah, so your odds really shifted once you have that other piece of additional information. Yeah.
So just the wording on these, to have a normal child was 1/24th, and then the probability for the second child to have the disease is also 1/24th.
This original probability was a child with the disease also.
Okay.
So I'm giving you, this isn't a probability here, this is just a new piece of information that they, suddenly they have a child, a normal child. You're given a new piece, if you were a genetic counselor, you're given a new piece of information that, okay, they had a normal child. Now it's a probability their second child will have the disease.
That one is their child had the disease, what is it?
Right, now I'm saying, okay, now they have a second child with the disease, what's the probability that a third child, or fourth, or fifth, or whatever,
Having the
has the disease, yeah. Any other? Is this problem, because I ran out of, we could start getting more inbred type things over here, but does it make sense how we're approaching this? Do you have kind of a sense of how to start with a problem like this? Definitely start by recording the information that you know, and especially if you're given the inheritance pattern of the disease, start assigning alleles to those individuals that you know the information before you even look at the first. Well, maybe look at the first question and make sure it's not anything unrelated to assigning all these genotypes. But if it's a probability problem, pedigree problem, that's your first step. Okay. So that was the end of our chapter 2. So that's the very end part of chapter 2, talks about pedigree analysis and chi-square, which we're going to get to. Before we get to the chi-square analysis, though, we're going to start in on chapter 6 information, which is about linkage. Okay, so linkage mapping, linkage analysis, and we're going to use chi-square to answer that. But you can also use chi-square for a lot of different hypotheses. This idea of linkage. So this is an exception to Mendel's, what he discovered, of course. And it was years, I think I told you guys that many years after Mendel did his work was rediscovered, finally accepted as the norm. Other geneticists started sort of popping up in the history of genetics. And Bateson, and you guys' favorite person, Reginald Punnett, who I know you all want to thank very much for giving you these lovely squares that you can draw over and over. So two individuals, Bateson and Punnett, this was in the early 1900s. Okay. They started seeing ratios that did not match Mendel's predictions, okay? So they found exceptions. To Mendel's predicted ratios, especially for the dihybrid crosses. So all of a sudden, a dihybrid cell, so two genes, heterozyte for both, wasn't producing a 9 to 3 to 3 to 1. Okay, so this was a clue for Bateson and Punnett that something weird was going on. Okay, so they were one of the first folks to find a dihybrid cross. Okay, that did not equal this 3 to 3 to 1, 9 to 3 to 3 to 1. And then a fellow who worked on flies, Thomas Morgan, okay, came out, and he worked on Drosophila, as I just said. And he's the one that actually showed that certain combinations of genes seem to appear more frequently. Okay, so he was the first, Morgan was the first person to actually show linkage. He also, almost all the Drosophila, original early data on Drosophila is from Morgan. Okay, he was pretty amazing. So he, of course, received the Nobel Prize. And he did all this work to show sex linkage that we talked about, the wide-eyed and red-eyed flies. That's from Morgan's work. Okay, so he used these fruit flies to show gene linkage. So genes that were traveling together and they were not assorting independently the way we would predict. And he did a lot of like really pivotal work, mostly at Columbia, and then he also came here to California. So he was one of the founders for Caltech, the biology program at Caltech. And I had the pleasure of taking a class actually from one of his, I guess, students or mentees named Mel Green, who I believe is still alive at UC Davis, also works on flies. And Mel Green was in his late 80s when I was in the PhD program there, and one of the classes we had to take was a history of genetics. And he would pretty much come into the classroom and sleep the entire class, and then randomly wake up and tell them the most amazing stories. And he had these big glasses that were like that thick of his eyes, and he was just amazing. And he knew Morgan personally, so he worked with Morgan and could tell all these stories to us about this phenomenal researcher and how he changed genetics. And Mel Green was a pretty awesome guy. He is a pretty awesome guy. But he did wake up at one point in the class and say, if you're doing statistics, if you have to do statistics and it isn't real data, and like everybody in the room as newly minted PhD candidates, you know, the idea that you don't have to do statistics to prove data, I mean, to all of you, that should be a pretty shocking statement. But back then, if you picture Morgan and these guys counting flies and just showing this real hard data with pure numbers, they're just showing out of 1,000 flies, we see a 9 to 3 to 3 to 1, or we see a 1 to 1 to 1. You don't have to do statistics. You don't even have to calculate an average to prove that. So for his context of research and science, that was legitimate. But you had a lot of shocked people in the room, like, oh, can I quote you? And it's not like, yeah, that. So that was Morgan and Mel Green, another person. For us to talk about Morgan's data, okay, and how he showed linkage, I realized that we didn't. Your textbook has an example and has gone through it, but we haven't really talked about what a test cross is, and I just want to make sure that we are on the same page for that. Okay, so that's what Morgan did, and it was really crucial to his findings. The fact that he did do a test cross is how he got the ratios that he saw. So what does that mean to people? If I say test cross, what do you think of recrossing? Yeah, so one, you're mating two individuals to figure out something about their genotype, something about the phenotypes. Someone over here said a heterozygote. Okay, so you're really, a test cross is you're mating a dihybrid, right? Heterozygote times what, though? By another heterozygote? Opposite. So homozygote recessive, yeah. So you're gonna mate them to a fully recessive individual. Okay, so that's a test cross. And we refer to this homozygote recessive individual as the tester parent. And the reason we do this with the homozygote recessive is in a typical dominance inheritance pattern. We're not talking about the extensions and epistasis and all these other crazy things. In your classic dominant recessive pattern of inheritance, the homozygote recessive phenotype, right, is easy to track. Okay, they're going to show that recessive phenotype. So basically, you're taking an individual that shows the recessive phenotype, and you're mating it to an individual that shows the dominant phenotype. And the reason that it's called a test cross is typically you don't know the genotype of the dihybrid. Okay, if we have a typical or classic classic dominant recessive inheritance pattern for a 2-gene trait in the phenotype, right? We know exactly what this individual, the tester parent's genotype is, but we don't know the dihybrid, right? I'm calling it a dihybrid, which means it's a heterozygote. But all you know in this case is that it's a dominant phenotype. So the reason they call this a test cross is you're trying to determine whether they're dominant, homozygous dominant, or whether they're heterozygous. So this is really, you're trying to figure out what the genotype is here, based on the progeny that you see. So really, and I want you guys to have this in your notes, is you're doing an analysis. You're tracking meiosis, right? You're going to track gametes, but your analysis can focus on only one parent. So only one meiotic outcome or event. So whereas all the examples where we've gone to the F1 and it's heterozygote and we're selfing that heterozygote, now you have two parents, two sets of meiosis events that are happening that you need to follow and track for gametes, right? But in a test cross, so an F1 dihybrid cross between two heterozygotes, you have to track two parents. Whereas if we do a test cross, we can essentially ignore the tester parents' alleles, right? Because they don't contribute anything. We know exactly what they're going to contribute every time. There's only one possible, right, gamete that they can ever give. Whereas the other parent, so your Punnett square is going to look kind of funky, right? Because the other parent, if they're a heterozygote, they can contribute all the sets of alleles that you guys are used to calculating, right? So what's our ratio we expect to see from this kind of cross? If the unknown is a heterozygote individual. Yeah, 1 to 1 to 1. They're all equally likely to end up with this 1 to 1 to 1 to 1 ratio. So that's a test cross. I just want to make sure we were on the same page for what that means. And that's what Morgan did. Okay. So any questions about the test cross? Does that make sense for? And everyone understands how you're really only tracking the meiosis, the gametes from that heterozygote parent. Okay. Okay, so Morgan saw a deviation from this 1 to 1 to 1, okay? So you used Drosophila, and we have some new allele designations. So there's eye color. The PR gene is for eye color. PR is recessive purple. The wild type PR plus is red eyes. And he looked at this is eye color. And he looked at a wing phenotype that determines the length of the wings. So the recessive is called vestigial. And the wild type wing is a normal, longer wing. Vestigial is like a little short, funky kind of wing. So, wing length. So these are the two genes I think that we'll start with. Has anyone actually worked with Drosophila ever? Yeah, it's actually really fun. You can just put them on a plate and pump CO2 through it, and they all go to sleep, and you can shuffle them around and throw them in new containers, and they mate and produce progeny. Until the CO2 tank goes out, which happened I did a lab for a period of time at UC Davis that we worked on flies, and So there was always flies like randomly in the room, or you'd be sitting on your dissecting scope with 200 flies that you've sorted out, and you feel a sneeze coming on. And that was like the worst thing ever, because about 4 hours of work of sorting flies through a microscope would be lost if you blew them everywhere. So yeah. Okay. Eye color, wing length. So these are the gene designation. And so what Morgan did is he had his parental generation, okay? So he made it a homozygous recessive fly to a homozygous dominant fly. And his F1 is, as we would expect, heterozygous for both traits, for the eye color and for wing length. So we got a dihybrid on his F1. But at the time, of course, he didn't know this, so I'm writing out genotypes. But really, all he's looking at here is a red-eyed, normal wing length fly. And he wants to know what its genotype is, right? So he's going to do a test cross. So he's going to cross this red-eyed normal wing to a purple-eyed vestigial wing fly. So this fully homozygous recessive fly and this particular example is our tester, this is our tester parent to produce our F2. So he crossed this F1, one of these F1 progeny, which is all red eye, normal wing length, to a purple vestigial wing fly. And got some data for the F2. So first, before I give you guys his actual numbers for the data, what do we expect to see from this cross? So what's our expectations for the F2 if everything is following our normal pattern? This is our test cross that we're doing right here between a dihybrid and the tester parent. Right, we expect a 1 to 1 to 1 to 1, right? So you guys should be able to, again, you check this homozygous recessive parent, this purple vestigial wing fly, you know the only gametes they can give are those two recessive alleles, right? And then the other parent gives all the combinations that we're used to solving. So these are the possible gametes. Okay, so we expect to see and I'm going to take the time, I know, for those of you who already have this, you can fall asleep. But for those of you who would like this in your notes and are lost at all, then here's our full Punnett square. Okay. So we expect to see in terms of phenotype, what are we looking at here? We're looking at a red-eyed, normal winged fly, and then a red-eyed, vestigial winged fly.
Purple. Oh wait, never mind.
And then two purple-eyed flies. Okay, one that has the normal wings and one that has the recessive vestigial wings. And we're seeing these in a 1-to-1-to-1 ratio, equal probability that each one of these phenotypic classes can occur. Okay. So that's what we would expect to see. Here's Morgan's results. Okay, so for the wild type gametes, so for those wild type alleles, right? So this is just looking at gametes, which can be inferred by the F2 progeny, okay? You got 1,339 lives. Okay, and then for the recessive combination of gametes, you got 1,195 flies. Okay, so this man spent a lot of time with flies. And then for the two heterozygote combinations of alleles for gametes, okay. So one with a recessive allele and one with a dominant allele, you got almost equal numbers to each other, 151 and 154. So kind of right off the bat of looking at those numbers, that data set, what do you think? Are we matching our 1-to-1-to-1 expected? Not so much, right? There's a huge overrepresentation. Okay, there's a huge overrepresentation of these two combinations. So wild type staying together and then the recessive mutant alleles staying together. So those tend to show up much more frequently. And so this is what Morgan was looking at, and of course he went on to figure out that what it meant is you have two genes that are actually linked. They tend to assort together, okay? And a way to kind of, for us to think about this, if you have two, so we're thinking about meiosis, right? And if we have two chromosome types, so if we're trying to visualize what's happening between two genes that are linked during meiosis, you have an R gene and an H gene. And what we're talking about is gametes and alleles that are homozygous together, or dominant together. So both of these individuals are homozygous, either dominant or homozygous recessive. Okay, so they're gametes. If we have a crossover occur here, not involving the alleles of the genes that we're thinking about. Okay, obviously there's other genes at the top of these chromosomal arms, but we're not concerned about them for this particular example. We could draw these cells after meiosis. You guys are all experts now, I know. Right? We could draw this after meiosis, and you should see your chromosomes all split up with their RH recessive alleles together, so little r, little h together, big R, big H together. Okay. So this is a normal, what we've been talking about. We haven't been considering any crossing over occurring between the alleles, right? Whereas if you were to take this same example and think about the other scenario, okay, where you actually have a crossover that occurs somehow that spans the location of the R and the H genes, and you draw the resulting cells after meiosis, now all of a sudden we see different combinations. We still have our one sister chromatid that has big R, big H for both of these chromosomes, okay? and then little r, little h. But then all of a sudden we have this chunk of, see if I can actually draw a little green bottom on this one. Okay, so now all of a sudden you have different combinations of alleles that we hadn't previously seen in the parental type. If the crossover happens between those two genes, okay? And then the key thing here is these events, okay, are less likely to occur. Actually, having a crossover between two genes, between these two alleles, is less likely to happen, especially the closer and closer those two genes are. We're going to talk about how you can use that information to map. Okay, so another word for crossover, right, is recombination, right? This is a form of genetic recombination. So that's why these two possibilities of meiosis, the bottom two cells up here that have recombined alleles, ones that we don't see in what we call the parental type, okay, we refer to these as recombinants. So the recombinants are a result, a product of that recombination, that crossover event. Okay, so this is how you get big R with little h and little r with big H. Okay? Whereas the parental groups, what we call parental gametes, what we see in the parents, okay, those are when our dominant alleles in this example stay together and the recessive alleles stay together. So that's what we're talking about when we think about genetic linkage and the fact that you can have crossover and recombination occur between two alleles that are staying, normally that are staying together and displaying linkage. We can separate those alleles out. And I would definitely my advice is to not get hung up with the trap that you always have uppercase letters together, dominant alleles together, you always have lowercase recessive alleles together. It totally depends on what the parental type is, right? So if your parent, if I had drawn these parents as not homozygote for these genes, but heterozygote, okay, you could have totally different combinations, right? So if this individual was heterozygote, they're both heterozygote, we could see now homozygote dominant alleles coming together in a recombinant progeny. Does that last part make sense to everybody?
So in that case, would the recombinant be more likely because they're attracted to each other?
Yeah, not based on any kind of principle like that, yeah. So always recombinants are more rare.
Okay.
That's a set, regardless of the allele designations, yeah, recombinations always between, if you're asking it for it to be between these two genes, is a more rare event.
Okay.
Yeah. There are areas on the genome that for different organisms, for different species, that they found recombination is really likely and happens at high occurrence. But we're not considering that piece for this class. But there are areas where, for whatever reason, as you kind of use the word, they're attracted or whatever, and that's a hot area of research right now, is trying to understand why in certain species, certain chromosomes have a ton of recombination occurring between alleles. And of course, that lends itself toward a lot of diversity in the progeny, which could be potentially evolutionary advantage or something along those lines. Yeah. But for this purposes, consider recombinants as being a more rare event. The likelihood of a crossover occurring between the two genes that you're particularly looking at is going to be less likely to happen. So we tend to see parental types in higher numbers than we do recombinants. Okay. All right. So how can we use chi-square, so we looked at this data set, and you guys said, nope, not 1 to 1 to 1. Okay, just based on how big of a skew we have, whole orders of magnitude. We have 1,000 progeny here and 100 progeny here. So the way to actually converse to what Mel Green taught me, that you don't need statistics to prove data, I'm telling you, you do. So to prove that, to actually show mathematically that yes, this is not following Mendelian inheritance, this is some sort of linkage or other inheritance pattern, we would use chi-square. Okay, so this idea of what chi-square is. Okay. One of the ways you can use chi-square is to statistically analyze this data set. Okay? So you're testing how true or valid is another way to think about it, your genetic hypothesis is. So we're looking right now, we're looking at this data set, and you guys are saying it's definitely not, here's sort of the ideas, the hypotheses. It's not a 1-to-1-to-1 that we would expect from a test cross. And we're kind of thinking that it might be linkage because, gee, that's the topic that we're talking about tonight in lecture. This is sort of what you're starting to think about, and we're going to use chi-square to address that. So you're merging all statistics and mathematics and then your genetic data to make some kind of more stringent assumption or conclusion about the data. Okay? So chi-square is a form, it's considered a form of hypothesis testing. Okay, because we're looking at how valid or true our predictions are. Chi-square is also, you'll see it referred to as a goodness of fit, if you've ever heard that term. And so certainly chi-square is used in many other fields besides genetics. This is not a genetic only, this is a statistical measure. You can use it in all kinds of data sets. If you go take stats from John Depka, he'll probably have a whole other monster to talk about with chi-square. I just want to make sure you guys understand chi-square as it relates to this type of genetic testing. Okay, so he might even use different words. It's really, statisticians are very particular about the wording that you use because it makes a difference in your predictions. Biologists are much more squishy fluffy with our wording for stats, that's for sure. Okay, so you're finding a goodness of fit. And you're finding a goodness of fit between so this measurement, I'll put a little star here so we can kind of bring our attention down here and show enough room for it. But what you're finding, that how good does your observed data fit your predicted data, okay? So this idea of a goodness of fit is between what you actually see, what you actually observe, and what your expected or predicted data is. So expected is another synonym for predicted. And your predicted data is what you base your null hypothesis on. So I know some of you guys are actually in my 242 class too, and we had an assignment in lab where you guys had to come up with a null hypothesis. So this term, and I know some of you had statistics before and some haven't, so we're a little bit all over the place with this. But this idea of having a null hypothesis, okay, you base your null hypothesis on the expected or predicted data, not observed data. So that's a key because that'll change how you analyze your chi-square, or how you conclude your chi-square analysis. So the decision you make at the end whether to accept or reject is you're deciding after you run this analysis whether to accept or reject your null hypothesis. So if you base your null hypothesis off of the wrong data set, you can actually essentially come up with the exact opposite answer of what you should have. Okay. So we'll spend some time talking about this. Okay. So in the case of, and we can put that up on the board so we can follow with this example. So we're using, here we were looking at this data set. This is our observed results, right? This is our observed data. Right? Our predicted was what? The 1 to 1 to 1, right? So our predicted or expected, which is what our null hypothesis is going to be based off of. And you'll see this case, I forget here. H with the subscript O, that's a null hypothesis. So you might see null hypothesis written as HO. So essentially our null hypothesis here is what? A 1 to 1 to 1, which is what? What does that mean?
Linkage?
No.
Between that and your data.
Well, so if our data is linkage, we'll just play like the what if game, right? And then that means our predicted or expected results, what we normally would expect to see if there were no linkage, is which one of Mendel's principles? Which of his laws? Independent assortment. This is independent. 1 to 1 to 1 in a test cross. That your null hypothesis in this case is independent assortment in Mendel's laws. It's following a Mendelian inheritance pattern. There's no deviation from Mendel's expected. Okay. Linkage would be the deviation. So linkage would be your HA if you're familiar with HOHA. That would be your alternate hypothesis. So the null hypothesis, which is based off our predicted data, our expected data. So this is the kind of the key, because chi-square is super easy to calculate. Okay, chi-square is not a difficult thing to calculate. You guys totally will get that. Where you get mixed up, where we see students get confused, is how to set up your hypotheses correctly so that you're testing the right hypothesis, right? Because if you don't, if you give yourself the wrong null, so in this example, if you had said my null hypothesis is linkage, then you're gonna set it up wrong. And you'll test the wrong thing and you'll get the wrong answer. The rest of it's easy peasy. So I would spend time with chi-square when you go to ASAP, when you come to my office hours, focus on how to find the right hypothesis for it. Okay? That's the piece that's difficult to see. Okay, so we're using chi-square to decide whether it's independent assortment or linkage is what we're seeing in our observed data there. Okay, and the formula is super easy. So chi-square is designated as Greek letter chi squared, and the formula is the sum, right, of your observed data points minus your expected data points. Okay, and then you square that value and you divide by your expected values. Okay. And again, your expected, these values we calculate, you have to figure out what your expected values are based on your null hypothesis. Okay. So our null hypothesis is that they're independently assorting. So have a set of numbers here. And we have this hypothesis that they're independently assorting. So how are we going to calculate this expected value? And this will change depending on what your hypothesis is, but in this particular example for linkage, we're predicting 1 to 1 to 1. So how do we get that?
Forms of the total views.
Yeah, so you take the total number of plants, total number of flies, total number of bunny rabbits, total number of zombies, whatever it is that you are calculating, you have to take that population that you're looking at, because you're trying to make predictions about this total population, okay? You're trying to make some kind of prediction about it. And our total, I wrote it down because I actually didn't feel like doing that math. Somewhere. 2,839 flies. Okay. So if we're predicting a 1 to 1 to 1 to 1, we're going to take that 2,839, and as Carl says, we're going to divide it by 4. Because we're expecting equal chunks, equal sets of flies in every one of those categories. Okay? So you end up with 709.75, but we can round this up to make it even more fun. So this is your expected value for each one of the points in this example. Okay? Now, if we did a different example where we were predicting, say it's a dihybrid cross with 2 heterozygotes, what's your ratio you would expect? A 9 to 3 to 3 to 1. So you would have to calculate 9/16, 3/16, 3/16, and 1/16 out of this total. Okay? So how you calculate your expected is based on your hypothesis. And in this case, we're hypothesizing equal portions for each of the 4 possible ratios, right? So your data is going to look like you're going to take each one of these sets of numbers. Okay, so 1339 is your observed. And you're going to minus what your expected is, which is our 710 value. Square that, divide by 710, etc. You're going to add up all these. We'll do the same. So 1195. So the squaring gets rid of any negative numbers you might have. Okay, so we add all this up. We do the math. This part's easy. Yes, you will have calculators on your exam before you start sweating that. Yeah.
For the 9,331, an easy way to do that would you divide by 16 and then times by 9 times by 3 times by 3.
Mathematically, you can figure it out however you want. I would probably just take 9/16 and multiply by.
Just multiply.
But yeah, whatever, however you'll have a calculator, so you guys can run the numbers. Yeah. So I think those are the tricky parts to chi-square, is making sure you have the right null hypothesis, and then making sure you calculate these expected values correctly. Okay, because obviously if you have your expected values wrong, if you have your null hypothesis wrong, all of a sudden the whole test kind of falls through the cracks there. Okay, so, and then I'll finish so we can see what the actual predictions are. So you end up with this value, right? You do all this multiplying and adding, dividing, square everything up, and you get, in this particular example, I wish I could do that math in my head. If I could, I probably wouldn't be standing here teaching. Okay, so we end up with 1,797. So I'll put approximately. Okay. So you have a table in your textbook, and you will have this on an exam. You'll have the chi-square table. Okay. You would not necessarily, in fact, you will not have that equation because it's the easiest question in the world to memorize. You guys can memorize this, okay? You'll have your calculator, you'll memorize that equation, you'll have, the chi-square table, so you can look up values on the chi-square table. Okay, and it's in your book, and it roughly looks like a table. Okay. So on the far left-hand column, there's something called degrees of freedom. And you might see it represented as a DF. So it goes down from 1 to, I don't know, 20 or something, I can't remember the full. I've never actually used that many degrees of freedom. And then coming across, so from left to right, there's a bunch of what we call p-values or probability values, okay? So these are likelihood values. So there's like, starts out with a p-value on the far left-hand side, typically, and some of these charts vary. So a 99%, I think it goes 95, etc., until we get all the way to the right-hand side, which has a 0.05 p-value and a 0.01. So generally, and this is where someone like John Netko would really have a lot more to say, but this is kind of tends to be your cutoff. Anything lower than 0.05. So if you end up with a p-value, these are all p-values here, probability values. And generally speaking, if you end up with a probability value of less than 0.05, you start to do what to your null hypothesis? You reject it, yeah. Okay. So if we, how this table is set up is it's saying that when you're down at this end, the 0.01, so you're saying that you have essentially a 1% chance or likelihood, probability that these numbers are happening by pure chance alone. So there's a 1 point, I won't use the word twice, so a 1% probability that your data set, whatever your numbers you're testing, are happening by chance alone. So that's the way to kind of think about this. So that, the result of that is that you reject. If you end up down at that 0.05 or 0.01, and I'll tell you what the cutoff is on an exam. I'll say using the 0.05 and lower, because some people want to be really stringent. It depends on the research and the data. Some folks will only accept the 0.01. They will not look at a 0.05. Some people push the other edge boundary of things. That's totally field specific and dependent on what you're looking at. But if we're in that 0.05, 0.01 range, you're rejecting your null hypothesis. Okay. So how do we find out whether we're going to reject that or not? So in the middle of this table, which I didn't fill in, for every degree of freedom, 1, 2, 3, 4, 5, 6, and each p-value, there's a number, okay? And so on this table, you're finding these numbers are the chi-square numbers, okay? So you would take this 1,797, and you're trying to find it on your table. The other piece you need is the degrees of freedom, right? You have to somehow calculate what degree of freedom you have. So to determine that, degrees of freedom are usually expressed as an n minus 1. Okay. So what's n? So n is usually described as the number of independent categories, okay? And this can get really tricky depending on the types of data sets you're working with. For you guys' case, it's going to be much more simplistic. But I'm giving you this generic definition so John Netko doesn't beat me to a pulp because I told you N actually just means phenotype of classes, because that's, if you go and use chi-square outside of genetics, you guys don't want to fall into that trap of them memorizing that, right? So in our case, degrees in this particular problem, our degrees of freedom are these phenotypic classes that were observed, and I erased our little chart there, right? So how many did we have?
4.
We have 4, right? So 4, our degrees of freedom is going to be 4 minus 1, so it's 3. So you would, that's why you would come over here, you would focus on the number 3 at your degrees of freedom, and you would read across this line until you encounter this number, or in the case of this number, it's so big, it's off the charts in this direction. I think this last number here at the 0.01 is like 20 or something like that. It's still many orders of magnitude, much less, smaller than 1,797. So you end up, for this particular example that we're running right now, you end up over here. Okay, so that's saying you have a 1% probability that you're seeing these numbers line up like this because of chance alone. So you're rejecting that independent assortment hypothesis. And that's where that key wording comes in, and John and other statisticians would definitely say, rejecting the null hypothesis does not prove that this is linkage, okay? All it says is that you're rejecting the hypothesis that they're independently assorting, but it's not a proof of linkage. Okay? It's probably leading us in that direction. We're thinking it's much more likely now because we rejected that they're independently assorting. But keep in mind that when you reject the null hypothesis, that does not prove your alternate hypothesis. You have to do further testing to determine that. Okay. So wait, before any questions about where we're at right now with chi-square? Okay. What I wanted to give you guys here, where did it go somewhere? I've heard a little bit from the ASAP tutors that people are already asking about chi-square, which is great. And again, I think the tricky piece is determining what your hypothesis is. If you're given a data set, trying to decide what your actual null hypothesis is, that's the hard part. And then making sure you calculate your expected values correctly. Okay. And in your homework set for chapter 2, I do not have any chi-square problems up there. In fact, this next homework set you have to turn in, there's only one problem due from chapter 2. And part of the reason of that is we're getting right near the exam, and I don't want to give you guys a ton of homework to do right before an exam when you're trying to study. But I know you need to practice chi-square. I know that that's going to be a helpful thing for you. So if those of you who are interested in chapter 2, there are a couple chi-square problems. They're all in the experimental E problem. So it's E10, E15, and E16. Those are all chi-square problems in your textbook. So you don't have to turn them into me. But if you want to use them as practice, I've already posted the solutions to them. They're up on iLearn. So all the solutions for the E experimental problems for chapter 2 is up on iLearn under this week's little section on the iLearn page. So you guys can work those problems and then check the solution, make sure you're still thinking correctly and approaching them probably. Obviously, come talk to me in office hours, talk to ASAP. But those are 3 additional chi-square problems that you guys can work on if you have 3 books.
But all of the chapter 2 homework is due next month.
There's only one problem from chapter.
She changed the syllabus like 2 weeks ago, I think.
Even on the original syllabus, there's only one chapter 2 problem. You had a whole, I'm sorry.
Yeah, no.
You're right. There's only one chapter 2 problem that's from this current
Yeah, there's still all the other
pedigree
chapter 2
stuff,
problems.
and it's a pedigree problem. But all the chapter 2 problems, but they don't include the
Okay.
chi-square.
I'm just having me confused.
Sorry, I misspoke. You're right.
I'm like, I did a lot of chapter 2 problems.
Yeah. No, don't also same, I said it before too, you guys, do as many of these homework problems as you can. Just send me an email if you're like, I wanna know what the solution is for it, and I can post solution manuals. No one's asked me for that, so I haven't been posting them. But if you guys want the solutions to problems that I'm not assigning because you're working them and you and the ASAP tutors aren't quite sure if you have the right, answer to it, just let me know. And I can post that. You guys, as much practice as you, there shouldn't be a mystery going into the exam. You should have had lots of practice working these problems, okay? Okay.
And actually, the new book one that you have doesn't have the first, doesn't have number 24 on it.
Oh, yeah, that was from fourth edition, so I can look up. But anywhere you see, if you have the fifth edition, so these are fourth edition problems. If you have fifth edition, just flip through, you guys can see right away if it's a chi-square. It'll say use chi-square analysis, to solve such and such, and then you can practice through that.
No, on the new book one you assign, it's completely not on that anymore.
Yeah, these ones?
The question 24 that you had this week.
Oh, yeah, I probably took it off because it wasn't a good question, yeah. Okay. All right, so any other questions about chi-square? Okay, so we still have a few minutes left, and what I want to do is just give you some intro notes to the next section, and then I'll let you guys head out of here tonight. So our very last thing that not very last, but for tonight, we're going to just introduce this linkage mapping concept. It ties together the ideas of linkage, okay? And you can also use, it'd be really hairy, but you could use chi-square too. Okay. Okay, so we know this idea of linkage, right? So we're talking about genetic recombination, right? Crossing over. We drew the chromosomes earlier showing results of a crossover that happened between two genes, okay? And what I wanted you guys to have in your notes here for this beginning is the greater the distance between those two genes, so the further and further apart that they fall on the chromosome. So if we had our little chromosome laying here sideways, I don't know why I always think about it sideways, but and you have genes A and B, and we had two possibilities maybe of how these genes are arranged. So maybe the top option, they're very far apart on the chromosome, and this bottom option, so option 1 and option 2 here, they're very close together. So between options 1 and 2, which is more likely to have a crossover, a recombination event occur between genes A and B?
One because two is too close to.
If you want to have DNA actually break and invade and recombine between two genes,
Yeah, two is too close.
think about how much space that takes. Yeah, it takes a ton of space to occur. So it fitting the DNA to actually break and recombine and invade the other homolog space between two genes that are very, very close together, this is an extremely rare event. So the bigger the distance, this option number 1, is much more likely to occur here. So we had said earlier, recombinants in general are rare. They're not very likely to happen. And on top of that, if two genes are very close together, it's next to impossible that you'll ever see a recombinant, a crossing over event occur in this option 2, where we have A and B very close together. So the greater the distance, the higher probability you have of a crossover happening between those two genes. The closer together they are, so in this case, closer together, so this one's more likely for a crossover event to occur between the genes. This second option, where they're very close together, it's more likely that you'll see them the alleles will segregate together. Okay? They will not be combined. Does that make sense? The fact that you need a lot of physical space to actually be there between the two genes to have recombination occur. Whereas if two genes are very, very close together, it's just so unlikely that the strands can even fit in that space, and all the recombination of proteins and nucleic acids to invade each other's physical domain. If two genes are very, very close together, you're gonna see them show up like linked genes more often. Yeah. Okay, everyone's completely zoning out now. I have homework still, so please come get your homework if you don't have it already, and I will see you guys on Wednesday.
Is it